Hello everyone. Welcome to today's webinar on Realizing Pharma DX Digital Classroom session. I am Vinto from Shimadzu Asia Pacific. Hereby after that I will refer it to as SAP, and I'll be your host today. In this session, we are going to discover Shimadzu's AI-driven solutions and automation for realizing Pharma DX. For those who are joining us first time, you may wonder what is Pharma DX. So it means Pharma Digital Transformation. To make the digital classrooms a smooth experience for everyone, please allow me to run through some housekeeping tips and console features which you can interact with throughout the digital classrooms. First, we have the share experience icon. You can click on the various social media icons to share these digital classrooms with your friends, followers, and connections on various social network pages. We also have some resources related to these digital classrooms available for free download. You can identify this resource icon at the bottom of your console panel. Just click on it and you can download the files. Lastly, I would like to point out that the Q&A widget allows you to post your questions. Just feel free to type your question into the chat box anytime during the digital classrooms and click on the submit button. Our speakers will try to answer as much questions as possible through the end of the digital classroom session. For today's digital classroom session, we have two exceptional speakers to introduce how to leverage Shimadzu AI and automation solutions to elevate your lab workflows effectively. At the end of the presentation, there will be a Q&A session where we will have the speakers answering your questions submitted in the Q&A chat box. So to start off with our first speaker, Wan Tong will be sharing about the AI features of Shimadzu AI series and Nexera series. Wan Tong is our Pharma and LC product manager at SAP. She has more than 15 years of working experience in analytical science and pharmaceutical industry. Prior to joining Shimadzu, Wan Tong has started her professional career as a QA/QC chemist in multinational pharmaceutical company. Over to you, Wan Tong.
Hi, good day to everyone. Thank you, Vinto, for your introduction. I'm Wan Tong, Shimadzu Pharma Market Manager. So today, I'm going to share with you one of the most important and interesting topics that we always discuss today is about the AI and automation. In the post-pandemic era, right, I believe all of you here agree with me that automation and AI is the hottest topic that we discuss every day. About the ChatGPT, we are using AI. AI is around us every day. So today, I want to discuss how Shimadzu AI, in our Shimadzu context, we call it the analytical intelligence, how it can accelerate your QA/QC lab productivity with our AI series. So Shimadzu LC lineup, we have two different types. One is the integrated type, which is on your left side, our AI series, and the other one is the modular type, the N series. But today, since majority of you here, you are from the Pharma QA/QC labs, so I would like to share with you more on the AI series, our Pharma QA/QC workhorse. AI series has come to the third generation. The new models now come with all the AI features that you can think of. You name it, we have it. We have the flow palette function, the IP finder to help you with your data processing. I believe that you all agree that data processing takes a lot of our daily time when we try to integrate the peaks, our chromatogram, so on and so forth. So how the IP finder and the IPDA can help you to ease your workload and to give you a peace of mind. And then, of course, the instrument. If today we have an instrument which can take care of itself, right? Why not? Then we can free our time to do more value-added tasks. And last but not least, we also release our Lab Solutions MD, which is for the method development solution for the analytical R&D lab. So this is also a method development software that come with the AI functions to help you with the method development workflow. Okay, so this is now finally, right, we have a HPLC system that is as smart as the human. We call it the living LC. The living LC can give us the efficiency as well as to maximize our throughput. So how does it help to maximize the lab throughput? First of all, we have the mobile phase monitoring function. This is just like you are having a balance on top of your LC system to measure the real-time volume of your mobile phase. So just in case, if you are running a very long batch sequence, you calculate the wrong volume of mobile phase that is required. So don't worry, this MPM device is there to help you, to tell you how much volume of mobile phase is required to complete this whole batch sequence. And we have the low flow pilot function. Flow pilot is to help you to increase your flow rate gradually so that when you run at a high flow rate or when you are using a small particle size column, your column will be well protected by the flow pilot function to ramp up the flow rate slowly. And it works in conjunction with the column oven temperature. And of course, right, in the pharma labs, if you do not want to expose yourself to the organic solvent all the time, you can access our system even from the office. You can control the system, the AI series from the office. Last but not least, we have the IPDA function. Maybe most of you will be thinking, what is IPDA? So the full name is Intelligent Peak Deconvolution Analysis. So this is the feature that can help you to separate the unseparated peaks, which I will share with you more later. And now the living LC, right, it can take care of itself by even telling the lab manager when the system is due for maintenance. And it will also preempt the manager whenever the system needs to change the consumable parts, right? It will also remind you. So this is how smart the living LC is. So it comes with the AI and automation function, including the intelligent startup and shutdown function. You can set at a preset timing when do you want the AI series to wake up and when to shut down. After you register your batch sequence, it will just shut down automatically. The mobile phase monitor, as I mentioned to you earlier, right, this is to help you to give you the real-time volume of the mobile phase required for the entire batch of sequence. The flow pilot is to protect your column. And the software also comes with a seamless method transfer program. Okay, this seamless method transfer program will help you to shift your method from a HPLC method to a UHPLC method, so on and so forth. And then iPeak Finder is to help you with the easy data integration. For the R&D labs, these are the two AI features that we want to share with you. One is to do the separation for the co-elution peak with the IPDA, as well as the peak intelligence, right, can give you the effortless peak integration. So now we move on to, as I believe most of our Pharma QA/QC users that you are online with us now, probably you already heard about this USP General Chapter 621 changes effective from December 2022. USP used to be very strict on the gradient change. However, after December 2022, right, you can see on my rightmost column here on this table, they have loosened the regulation to allow our Pharma QA/QC users to change your gradient method. So if you want to convert your existing conventional HPLC method to a UHPLC method with a smaller particle size column, you can do so. Just to make sure that you follow these criteria that's shown on this table, your column particle size must follow as per the length over particle size, right? When you do this equation, it must be minus 25% to plus 50%. If you can ensure that you follow 100% these criteria set by the USP, then the good news is here, the method modernization, as long as it is compliant with the USP General Chapter 621, you can be exempted from the revalidation. But having said that, right, if you need to convert to a UHPLC method, there are so many parameters that you need to take care of. Does Shimadzu have a solution to help you with this? Yes. Later, I will introduce to you. This is an example of the method modernization of one USP monograph. You can see on my screen here, in order to convert this method and make it a faster method, shorten it from 20 minutes runtime to 10 minutes runtime. These are all the calculations that you need to do to ensure that you follow the USP General Chapter 621 regulation. But if, let's say, today I have a program to help you to do all this calculation, are you happy with that? I think most of you will be happy with if there is this Shimadzu method transfer tool. So this transfer tool, right, it already comes with the L over DP calculation here. So if you have an existing HPLC conventional method in your lab that you are running and you are using now, you want to convert it to a fast method, you can just set on this program, the method transfer tool, to set what is the UHPLC column that you want to use. Then the software will automatically tell you whether you follow and ensure that you are within the criteria of the USP 621. L over DP must be within the range. Not only that, if you are converting a gradient method, if you have to calculate all by yourself, right, it is a very tedious process. So that's the reason why Shimadzu came out with this method transfer tool to ease your workload. Even if you have any other third-party vendor's system and you want to convert to a Shimadzu system method, right, you can also set accordingly in the configuration, right? What is the third-party vendor configuration, including the system delay volume, the mixer volume, and then this program will help you to convert directly from a third-party vendor system to a Shimadzu AI series or the N series method. So isn't this a good news? Yes, I believe you all are happy to use this program to help you with the method transfer. Okay, moving on. As I mentioned earlier, the system has the auto startup function. For example, if you start your work at 8 o'clock in the morning, you can set the system, the AI series, to automatically start up at 7:45 a.m. So by the time when you finish your breakfast, you had your coffee, you enter your lab, right? You can just put the standards and sample in the auto sampler and start the analysis immediately without having to wait for the conditioning of the system. And this is the MPM, the mobile phase monitoring function. So this is a patented function whereby now you have a balance on top of your AI series. So this MPM, right, is telling you how much is the real-time volume of your mobile phase. Then when you register your long batch of sequence, if your mobile phase that you prepare, right, is not sufficient to finish the entire batch of analysis, there is this information dialog that's shown on the lab solutions, right? It will pop up and tell you that, hey, the mobile phase that you prepare is not sufficient. please prepare more mobile phase, or either you can reduce some of the injections from your batch sequence. So this is the function, right, to help you to prevent your system dry out, to prevent your column damage. And finally, right, we don't want to introduce air bubbles into our system if the mobile phase is not sufficient. Yep. And some of you might be asking me, hey, Wan Tong, how about if, let's say, I'm using 5-liter mobile phase? Yes, no problem. We do have the 5-liter mobile phase bottle holder also. So you can mix and match according to the mobile phase bottle that you use. So we have different types of the mobile phase holder. We have 1-liter holder that you can put any bottles that is, you can fit in the holder, you can use it to calibrate, and then you can just use it to calculate the real-time volume of your mobile phase. If some of you are using the 5-liter bottle, no problem. Then you can just purchase the 5-liter holder. So in this 5-liter holder, right, you can put any 2-liter mobile phase inside also can. So this is giving you the real-time volume of your mobile phase, so that you will always make sure that you have sufficient mobile phase for your entire batch of analysis, especially for the gradient analysis, which it is very difficult for the chemist to calculate exactly how much volume is required when it comes to the gradient program. And the next feature, right, the next AI feature that I like to introduce is the flow pilot function. So Shimadzu flow pilot function, right, it works in conjunction with the column oven temperature. The conventional way is the chemists have to sit in front the instrument and increase the flow rate gradually. This is a way to protect your column, because we all know that column is not cheap also. Column is expensive and we want to protect your column. So now with the flow pilot function, right, you can see that this is with the flow pilot function. For example, your flow rate is 1 ml per minute. With this function, right, if let's say you set the flow pilot to be 2 minutes, the software will use like 1 minute to increase your flow rate from 0 to 0.5 ml per minute. Then it will wait for the column oven temperature to stabilize. And once it's stabilized, right, the system will automatically increase slowly from 0.5 ml per minute to 1 ml per minute flow rate. So this is also to extend the lifespan of your delicate HPLC column and to maximize your return on the investment, so that you don't have to always changing the column. Okay, and the next one is how to increase your lab throughput. With our auto sampler, now it has a very high sample capacity. Up to 216 2 ml vials can be put inside this rack. And even when the system is running, you can still pull out the auto sampler to put your sample inside for the subsequent injections. And you can also put different rack type in this auto sampler. For example, if I want to put 96 well plate, yes, you can also put inside. With the rack label, right, the auto sampler will automatically detect the correct rack for you with the special label. So it will auto identify what is this rack that you use, so that to prevent the human error. So this is very high throughput, sample throughput to increase your lab productivity. Moving on, I understand that in the Pharma QA/QC lab, your product is meant for the human health. So the reliability of the results is very important. With the AI series auto sampler, right, now it has the very robust auto sampler that can ensure ultra low carryover. For example, if you inject a very high concentration of sample or standards, right, followed by a blank, you can see on this screen that the carryover is very, very minimal, can achieve 0.0025% of the carryover. So this is to also ensure the excellent repeatability as well as to make sure that your results is reliable. AI series, it also comes with the column oven. Of course, column oven is required to store our column. So in the column oven, you can install up to 6 short columns and 3 long columns. So it has the wide and big column oven capacity, allows you to even install the switching valve so that you can switch among different methods automatically. This column oven, now we have extended the oven temperature to room temperature minus 15 degrees Celsius. So if you are analyzing some USB monograph that requires the oven temperature to be at 10 degrees Celsius, using this oven, right, with our low temperature oven option, you can achieve this temperature. And the system comes with a front panel in front of the system, as you can see on my screen now. So this front panel, right, allows you to set the parameters directly from the front panel. And in case, if let's say you want to do some troubleshooting on the system itself, right, even the end user, you can also just use your smart devices to scan the QR code on the screen and then to find out how to do the end user maintenance. For example, how to clean the suction filter of the mobile face, so on and so forth. You can find all this video from the front panel screen by just scanning the QR code on the screen. So also this front panel allows you, at one glance, you know what's going on to your analysis, whether is the system running or is the system in the standby mode. You can easily see from the screen here. And we also have the living algorithm. So how does this living algorithm and the automation helps you to ease your and simplify your workflow? First of all, you can see on the screen here, we have the automated sample pre-treatment function. So if you want to do auto dilution to replace the manual dilution, it is possible with our auto sampler. And the IP finder is sometimes there are some very challenging peaks or chromatograms with the API main peak as well as a small impurity peak. How do you integrate these peaks accordingly? And how do you integrate them correctly, right? You can use our IP finder. It's a new algorithm for the data processing. And we also have the seamless method transfer integrated in the lab solution CS. So if, let's say, you have some existing method in your Shimadzu LC and you want to convert it to a UHPLC method, right? This is the integrated program in the lab solutions that you can use for the seamless method transfer. And the IPDEA is to resolve the unresolved peak, like what is shown on the screen now, so that even if there is some co-elution, the IPDEA function can help you to separate the peaks. Okay, so this is the auto dilution that I mentioned in the auto sampler, can help you to do the dilution automatically, as well as if there are some application that you need to add the reagent, right? So this is the auto addition reagent that is available in our AI series auto sampler pre-treatment function. The co-agention simple mode is also allows you to mix with additional reagent. For example, in some application like the pre-column derivatization of amino acids, you have to add multiple reagents in order to derivatize the amino acids. So in the auto sampler pre-treatment function, right, you can program the needle in such a way that to add a few reagents to derivatize your amino acids, and then it will mix within the needle and the sample loop. And then this is the result that you can get for the online pre-column derivatization of 20 amino acids. Because, right, if you derivatize the amino acid manually and you didn't inject into the column immediately, the derivatized sample will tend to degrade. So, but with the full automation, right, it ensures that after derivatization done by the needle, immediately it will just inject into the column. And this all can be pre-programmed. So if you want to understand more about our auto sampler pre-treatment function, right, feel free to contact your local Shimadzu sales representative. They are more than happy to share with you. And this is the iPeak Finder function that helps you to integrate your peak in a very consistent way. For example, if you can see some examples here, right? If you have a shoulder peak to your main peak, what is the correct integration and how to make it consistent? You can set easily on our iPeak Finder a new algorithm for the data integration on lab solutions that can make sure that you have the consistent peak integration using this new algorithm. And method transfer, as I mentioned earlier, now everybody is talking about green revolution. We want sustainability. How to reduce your organic solvent consumption? One way is to convert your conventional HPLC method to a UHPLC method, make it faster, make it shorter runtime so that you can use less organic solvent. But when you are talking about the method transfer, right, there are so many parameters that you need to consider: flow rate, sampling rate, system delay volume, injection volume, gradient program. So now we have the integrated method transfer function that can help you to convert the method seamlessly in the software of Lab Solutions, Shimadzu Lab Solutions. So you can just easily click this method transfer button, open your existing method, the HPLC method, then you can key in the new column dimension that you would like to use, the UHPLC column dimension, and you just need to click the button convert, and the method will just convert seamlessly for you to the UHPLC method, including the gradient program. Then you can save it as a new method file. So this is to help you to convert seamlessly between the HPLC method and the UHPLC method, vice versa. Now I want to give you an example of the method transfer of antibiotics. To convert from a 40 minutes method, shorten it by just a few clicks. You can shorten to a UHPLC method with a faster analysis time. So this can reduce your analysis time by 33%. Okay, so it can help you. Now you only requires like 33% of the analysis time compared to last time when you have a conventional method which requires like 40 minutes. Now you have shortened it to 12 minutes using the integrated method transfer program from Lab Solutions. Okay, so moving on, this is the IPDA functions. Sometimes, right, we analyze the main API, the active pharma ingredients, but we also need to check whether there is any co-elution with the impurity. So if you suspect that there is some co-elution in your main peak, right, you can use the IPDA function to check. This is to confirm that, oh, in fact, there is a co-elution here. And not only the software helps you to separate the peak, but it also helps you to separate the UV spectra. So this function is available when you are using our PDA detector together with the Lab Solutions. Some of you might be wondering, how many peaks can be deconvolute? How many peaks can be separated by using the IPDA function? The answer is up to 5 peaks can be separated using this function. And this is a unique function to Shimadzu only, Shimadzu Lab Solutions software together with the PDA detector. Okay, so moving on, this is the Smart Automation Kit. In short, we call it the SA Kit to accelerate your method switching and the method development. So you can have the option of either 4 mobile phase or 7 mobile phase Smart Automation Kit. It can help you to switch among 6 different columns or 6 different methods. You can choose whether you want a 2-column switching or a 6-column switching. So this switching, right, can be operated automatically, and the time that required to change the column is significantly reduced because you do not have to be there at the system and manually change the column by yourself. You can use the auto switching valve. So this can also eliminate the human error because ultimately, right, you do not want to, like, for example, change to the wrong column. Everything can be programmed in the software. Okay, so this is all the automation to help you to reduce your workload because you do not have to always be there in the lab and you do not have to inhale all the organic solvent. This is to help you to stay away from the lab and you can do like more value-added work in the office rather than always expose yourself to the organic solvent in the lab. Okay, so for those like pharma R&D lab, if you need to do the method development, Shimadzu, we have the method development solution called the Lab Solutions MD. So this is an overlay software that work with our lab solutions that enable the AI and automation to shorten your method development time. And it also comes with the automated peak tracking function to save the time and the labor during the data processing. And it can achieve high efficiency so that you can reduce the time that is required for the method development. Okay, so AI series, again, it looks very compact, but in fact, it is very flexible and expandable. So you can see now, even if I want to add another additional detector, originally it already comes with either UV or PDA detector. But now, if I want to add an additional detector on top of the AI series, it is very flexible. You can still add like additional detector. If you want to analyze like the UV chromophores compound, as well as some compounds, right, they cannot absorb UV. You want to add additional detector like RID, refractive index detector, or even MS, the mass spec also possible with our AI series. Okay, so yes, maybe most of you here from the Pharma QA/QC labs, the users, not only you have Shimadzu system, you also have like other vendor system. So the data can be easily convert from other vendor system or some Shimadzu legacy LC system, right, to the AI series. Now I want to coming to the end of my sharing here. So the analytical intelligence, right, brings the work flexibility. So it allows you to work from anywhere. Yep. And this is the summary. Finally, now we have the living LC that is as smart as human, as smart as our audience here. So this is to help you to really take care, the system can take care of itself so that you do not have to spend so much time troubleshooting the system, because now everything, it will just take care by itself. And then with the value addition of the software feature, including the IP Finder, IPDEA, it can help you to ease your workflow. And it comes with all the AI and the automation workflow to help you to increase your efficiency and improve your lab productivity. And this is just to thank all our users that you are online with me now. This is our celebration last year in India. We have sold like more than 10,000 in India. So next year, we also have another big celebration for our AI series support. We will not come this far without your support. And finally, today I'm sharing with you most on the QA/QC features. But if let's say you have other, like you are from drug discovery or you are from the clinical trials lab, BABE labs, right? Shimadzu, we offer the entire workflow for the pharma segments from drug discovery to the pharma QA/QC testing. And with our informatics solutions. Now, again, our entire workflow, right, is powered by the Living Laboratory. It is a Shimadzu ecosystem, which our next speaker, Dr. Kani will share with you more. Thank you so much for your attention, and that's all for my sharing. Back to you, Vinto.
Hi guys, thank you for Fan Tong for sharing a wonderful presentation on how the AI features of Shimadzu AI series and Nexera series to help with realization of Pharma DX for the users. Our next speaker, Dr. Kani, will be talking about the pathways to establish sustainable compliance environment by Pharma DX with Shimadzu's AI and automation solutions. Dr. Kani is our informatics manager at SAP, has 10 years of experience in laboratory management and informatics systems, bringing solutions to your needs in lab informatics, such as the client-server network systems, lab solutions CS, LIMS, other QMS applications, and more for streamlining lab workflows, regulatory compliance requirements for electronic data management, and lab practices and analysis. Over to you, Dr. Kani.
Thank you, Vinto. Hello, everyone. So far, you heard from my colleague Wantong about how the Shimadzu's AI features of our instruments automate your lab productivity and analysis. Now, it's time to learn about the digital transformation with Shimadzu solutions. As you all know, this in the digitalization era, we all aware that digitalization is very important. Transform our manual practice to digital is the need of the hour. But how we can approach the digital transformation, especially for pharma manufacturing, adopting agility for digitalization is the key for the successful transformation. That is how we discuss this session of my sharing. With that note, I wanted to give a recap of this Pharma 4.0, which is strongly recommended by ISPE to adopt the predictive analysis, data analytics, connectivity, and computerization to achieve the automation integration of this manufacturing practices. And with this, we can achieve compliance, more importantly, and a huge productivity and efficiency. For that, we need to understand what are the various processes that we have in a pharmaceutical quality. In pharmaceutical manufacturing, we have a segment of processes in QA, in QC, and production, and also for the various departments like document control, training management. And all these quality practices need to be very compliant way, and it should be a procedural, and it has to be a control manner. The entire process very tedious to maintain manually for long. When the companies or the market demand for the production is increases, these quality practices need to be stringent and also very control manner for the efficiency and productivity to meet the production demands. How we can achieve with the digital transformation. This is my sharing today. How Shimadzu's the total solutions, end-to-end integrated quality systems can automate all these quality practices of this pharma manufacturing. We start with this Lab Solution CS, our client server network system, Lab Solution BIX, our analytics, and the complete quality system, starting from LIMS, quality assurance management system, document management system, training management system, and BRM, production batch record management for the production systems, and more importantly, the APQR, annual product quality review reports. The pharma quality procedures, whatever we've seen in the previous slide, all can be covered with all the solutions. Most importantly, it is integrated to each other to give the complete automation. How to approach this complete automation? To understand that, we need to know how the digital transformation helping the laboratories first. That is the first step of automation. First thing is the compliance. If we have a digital control workflow, it will be it ensures the compliance. And importantly, the manual practices are no longer sufficient to adhere the compliance. And it is very difficult to maintain the environment and sustainable way. That is how we have to change our practices in a digital control manner, and it gives us productivity. To meet the increasing demand in pharmaceutical production, digitalization is inevitable, and it is the only way to maintain the system effortlessly and comfortable way. And this also paves the way to adopt the new technologies. For example, in a conventional way, the integration options are less and data transfers are impossible. To adhere the advancement in the technology, we have to change our platform to digital. That is how, without digital transformation, we cannot adopt the latest technologies such as cloud or remote access or integrations, which is very important for the automation. When we talk about that, we also need to think in compliant environment, what regulatory body is keen to inspect. There are two aspects in terms of laboratory. One is reliability of the analysis data. Upon the generation of electronic data, it should be integrity and security, audit trails, e-signatures, backups, validations. These are all need to be ensured. And second part is the sample process, reliability of the entire sample process, starting from how the sample is handled and relevance of the test operations and judgment accuracy and organization health, approvals, review procedures. All these are taken to consider for the compliant, to ensure the compliance. How this can be achieved? The analysis reliability can be ensured with the complete data management system. And the sample process reliability will be ensured by the comprehensive sample management system. When we talk about the data management system, it should be a backbone of the laboratory. In a conventional terms, we call it a CDS because it's a chromatographic data system. But our lab solution CS is the analytical data system. It's ADS. It not only giving the data management security and integrity, it is acting as a backbone of the data management by covering the entire electronic data management in a centralized way. It also ensures the accuracy, consistency, traceability, and transparency of the data. As you can see here in this high-level diagram, a centralized server can connect any instruments in the laboratory, and not only for the data management, it can provide the operational workflows for each and every instrument. It gives the advanced data acquisitions, results and processing options, reporting workflows. These are the main features that ensures the compliance with productivity. So in short, the manufacturers need robust and reliable data management system to guarantee and demonstrate the regulatory compliance. And one unique function of Lab Solution CS is that it can give the data management by entire instruments in the laboratory, not only Shimadzu instruments or chromatography instruments. It gives the spectroscopy instrument, non-Shimadzu instruments, and many more options for your complete data management. What are the value proposition with Lab Solution CS? This is the analytical data system to integrate every instrument in the laboratory. In short, Shimadzu is the only vendor can provide the balance to mass spec instrument connectivity. And many AI features for workflow automation ensures the efficiency and productivity, because these advanced features automate the operational workflows such as the data acquisition, processing and reporting, and many reporting tools that eliminate the transcriptions error, and it gives the automation. Very short time we can prepare all our reports in a compliant way. That is the second important feature of any client serverless system. And next is the network ensures the compliance, like adhering the compliance from the validation to the disaster recovery as per the regulations, not only FDA, any other regulations. That is the we can set up the compliant network in the laboratory. And next, as I mentioned, regulatory compliance is not only for the chromatography data, it's for the any instruments that data is impacting the final product quality and patient safety, right? Important to ensure the data integrity of non-chromatography instruments due to increasing warning letters issued related to the UV and IR data. And not only that, even a balanced data that has the impact in the final product quality. That's how Lab Solution CS can connect from the balance through the mass spectrometry into the single network. How it can be done? Because we do have many vendor instruments in the laboratory, non-Shimadzu instruments, many other instruments which file size is more and which cannot be handled by the normal system. We have a solution for that. Any third-party instruments can be controlled with Lab Solutions, direct control of Agilent and Thermo LCGCs. If you have any other systems, non-Shimadzu instruments, we have a very unique tool called MIDR, Multi-Instrument Data Registration. This will collect the results and PDF reports from any instrument and stored in the centralized server. We can make use of the data for reporting and any other purpose. And we also have a very unique backup tool, which can act as a watcher in any of the instrument that can collect the data and back up into the server folders. And this way, we can cover the entire lab data management with the single application, which is Lab Solution CS. What else it can give? It gives the laboratory analytics. Lab Solution has a unique function, which is a unique tool can be applied with Lab Solution, which is called Lab Solution BIX. Is the unique analytics. It gives the graphical dashboards from the information available in the laboratory about the instrument utilization, user performance, and audit trials, and lab performance, analysis planning or scheduling. There are many functions which gives the overview of the laboratory. And this is the web-based system. It can be implemented together with Lab Solutions for the interactive information about the laboratory. It gives the overview of the laboratory about how much the instruments are utilized, how many hours the instruments are run, and whether any instruments are underutilized, any training requirements based on the error and warning messages. It gives a complete overview of the laboratory. And moreover, it can calculate the number of batches and product released per month, which gives the lab performance as well. And with this, Lab Solution CS is the complete solution for the laboratory management as well as the data management of the laboratory with Lab Solution BIX. And second part of the digitalization in the laboratory is the automating the sample management. And as you all know, the sample management is a very detailed procedure. From the sample login to the COH generation, that certain workflow need to be followed to ensure the productivity as well as the compliance. How this can be achieved with a proper digital solution, we can say it's a LIMS, Laboratory Information Management System. Because a laboratory is all about the samples and sample-related processes. There are too many samples in the laboratory, and there are a lot of logbooks and verbal communications about the job assignment. And we do have many instruments that need to be managed, calibrations, preventive maintenance. There are many chemicals and inventories that need to be managed, stock checking and reconciliation. And our reporting process is very tedious for the manual calculation documentations. And also, there is a compilation of reporting and also review and approval process, which makes this manual process much tedious, need more manpower, and need more vigilance to make it compliant. And this LIMS application, which is a web-based software for the effective lab management. It can take care of the data related to the samples, experiments, workflows, instruments. Everything is digitalized in a centralized control workflow, which is a software-driven workflow. And a modern LIMS system can be a digital backbone of the laboratory that covers the entire process digitally. how modern LIMS can work. This is the sample life cycle from the sample login to the COA generation. When we log in the sample, this sample information can be come from the ERP systems. In a pharmaceutical manufacturing, ERP is a very common and it is inevitable as well. And many of us having the ERP systems. And LIMS take this sample information directly from the ERP systems and it can give the automation starting from the sample login itself. It avoids the logbooks and it gives the next process in a digital way. For example, any sampling procedures, it can be done digitally. And sample allotment in the application itself. Upon the sample allotment, the users can record the results in an electronic worksheet instead of any paper documents. And this electronic worksheet is a very unique, powerful tool which can capture the inventory information such as reference standards, chemicals, and instrument calibration status, and everything. And second, it also can capture the instrument results from any type of instruments in the laboratory. We do have a port type instruments and file type instruments like such as UV, TOC kind of instruments or CDS, network systems. The data from any instrument or any type of instruments, LIMS will handle in such a way that it captures the PDF reports directly from the instrument and it can capture the individual results for the calculations. In this way, we can avoid the transcriptions error and saves a lot of time for documentation, saves a lot of time for review and approval as well. And this review and approval can be done electronically by taking care of every piece of information about the analysis, and the COA will be generated upon approval. And this COA can be sent out to the any individual department which is taking care of the results. And also it can be pushed back to the ERP system. So the test results will be pushed to the ERP systems as well. In this way, the complete sample life cycle will be automated. And what is the advantage of having this kind of modern system? It is a web-based application and it is a completely plug and play modular system. What is the advantage? It's easy to implement. It's easy to scalable, meaning starting from the simple modules and then keep adding up the other integrations. And it's a complete automation with the Caliber LIMS. We no need to have any paper documents in any of the process in the laboratory. We do have many paperless laboratories with this application. It is already implemented. And it has a very unique features that inbuilt electronic worksheets and very pharma-specific workflows such as stage-wise testings and OS, OOT investigations. There are many modules that is curatively developed for the pharma industry. In short, it's a very pharma-specific LIMS application and ready-to-use workflow as well. There is no customization needed. We can just implement and prepare the configurations and start using it. This is the way to approach the digital transformation with the LIMS and data management. Here, the LIMS application can automate the sample workflow. It also gives the complete information about the sample process. That is the beauty of any digital system that can be done with the complete LIMS system. Now we come to know that how this can be digitalization done, but how to adopt the agility, how to start the system digital transformations. This is the digitalization roadmap with the Shimadzu solutions. We have a very scalable solutions starting with the simple package, which is we call iPackage, which is the startup data management solution for the any laboratory. It can connect up to 10 instruments into the network. It's very cost-effective server network for LCGC instrument. We can connect any number of spectroscopy instrument and other instruments in the system. It's fully functional and scalable, and it's completely compliant with regulatory such as 21 Part 11, CFR Part 11, UU or PICS, MHRA, any kind of regulations, we can set up the system as per the regulatory requirements. And this is the first step to make our digital journey in the laboratory data management. And LIMS, we do have the scalable LIMS application as well. Since the LIMS is a very comprehensive system, it's very tedious to implement in one shot. We have a roadmap for the LIMS application as well. The iLIMS is the entry-level application that requires affordable investment and shorter implementation time, and it encourages the digital maturity of the people so they can easily adopt this workflow very comfortably, can scalable later. And it requires less manpower for the implementation, so it will not impact the routine function of the laboratory. So small and medium companies have a huge concern to fulfill the above requirements to kickstart the digitalization. That can be done with the iLIMS. It's a very simple, cost-effective, but complete solution for sample management. And this is the suitable way to kickstart our digitalization for the LIMS. And anything that is we kickstart the simple package, the scalability should be there to attain the complete digitalization in future. When the companies grows, when we expand our production and business, this is the very important feature to have a scalable solution. This iLIMS can be upgraded to the standard LIMS, having the complete modules and integrations, and also can be implemented as an enterprise version to adopt the multiple sites in a single application. This way, Shimadzu solutions like iPackage or iLIMS can be expandable, scalable upon the need of the business. And this way, the integrated Shimadzu informatics can be implemented in a simple way, which can attain the complete paperless complaint environment as well. It's not only for the LIMS or our data management. We can do this kind of scalable digitalization for QA process or production system or much more quality matrices as well. With this note, I can say digital transformation allows us to give the work flexibility, which is work from anywhere or 2 days work week, and even weekend automation. As you heard from my colleague, that our instruments can give the automation in the analysis, our web-based systems and remote accessibility of our data management systems allows you to work from anywhere. It gives the comprehensive work flexibility. And thank you. And these solutions will be giving the very fastest ROI because this is all scalable. So once we implement, you can start improving your productivity. It automatically saves the time and gives the data access and increase the quality. Automatically, the ROI will be faster. And this is the way to prepare our laboratory for digital transformation for the better tomorrow. With this note, I thank all of you for hearing my session. Please post your questions in the chat box. We will answer your questions. Over to Vinto.
Hi, thank you, Dr. Kani, for your presentation. We hope that you have better clarity on how to make quality control more efficient with Shimadzu's data management and software. We will now move on to the Q&A session. Let us welcome back Wan Tong and Dr. Kani to answer your questions. Hello.
Hello.
We have some questions coming in, so please feel free to send in more questions so that we can answer them today. Now, I see that there are 3 questions available first, and let's have the... Okay, this is from Kishore Chaudhuri. So we have this question where the how to generate the HPLC gradient method of any product. So let's invite Wan Tong to answer this question.
Yes, yeah. So Vinto, I will answer this question. So actually, there are more questions coming in, but not to worry, even though now we overrun a little bit, but we will, for those unanswered questions, right, we can send to your email and answer your questions accordingly. So now let me answer this question from Kishore first. How to generate LC gradient method of any product? Okay, so basically, right, as I just now I introduced our Lab Solutions MD. It is a method development software. So this method development software, right, it comes with the AI gradient optimization function. So I believe you all agree with me that gradient optimization is the most tedious part in the method development workflow. So this AI function, right, it is so amazing that you just need to give a very simple one-step gradient program to the software. So based on your criteria, you can key in the criteria, for example, like what resolution you want to achieve. Majority of the USP monograph, you have to achieve like resolution of 1.5 or 2.0 and above. You can set the criteria, and you can also set other criteria like the tailing factor must be less than 1.5, this kind of criteria. And then the software Lab Solutions MD, right, it can come up with the gradient that is optimal for your separation of maybe like 12 peaks, 20 peaks, or 25 peaks. So this is the AI gradient optimization function. So if you want to generate the gradient method, right, I think Shimadzu can provide you best solution is the Lab Solutions MD, method development solution. So Kishore, I hope I answer your questions. So but Lab Solutions MD, right, if you are interested to understand this product more, right, please arrange an online demo with our local Shimadzu sales representative. We are more than happy to conduct an online demonstration for you. Okay, so please contact us. Yes. Yeah, Vinto, I think we also have questions for Kani, right? Yes.
Yeah, we have one question, I think, for Kani. Let us invite Kani to answer this question, which is how to bring non-PC based instruments to LIMS.
Yes. Yeah. It's a valid question because we talk about the digital data, electronic data. We all know that how this integration is possible, but being non-PC based instruments such as balance, conductivity meter, auto titrator, much other instruments, which currently having the printer is connected, which providing the paper output. And these instruments is very important to connect into the LIMS. The reason why, because these are all the paper documents, there is no traceability or audit trails or any validity of these documents. If the paper is lost, then the data integrity is lost. And that's how that Caliber LIMS has a very robust mechanism to connect with each and every port type instruments, directly connect into the LIMS server. We use a very small tool called, it's a device which is the analog to digital converters. These converters is a very robust way, can convert these analog signals from these balancers, and the digital values will be directly printed to the electronic worksheet. So when you keep the samples in the balance and print, it will automatically print into your electronic worksheet with all the information like which instrument, what is the instrument ID, when it was last calibrated, and who is the user using the instrument, date and time, and which sample, which test. All information is captured with no time, and this is giving the traceability and data integrity for the port type instruments as well. Did I answer your question? Hope this helps. Yeah.
Yeah, thank you, Dr. Kani. Hopefully the question is answered. If you have like further inquiries, of course, you can continue your question, but we will just wrap up in maybe one or two more questions. Then the further questions will be answered by email. So we have, okay, we just have two more questions, right? Okay, let's go through one of them. Maybe this one. So we have one question from Ani Patil. So please explain the method transfer tool if possible. So yeah, Wan Tong, over to you.
Okay, yes, Anil, thank you for this question. So first of all, right, maybe actually I can share again my slide earlier. Shimadzu, we have two different method transfer program cater for users. For example, the users is an existing user of Shimadzu LC system. We have this integrated method transfer program. It is already built in the lab solutions. Just now I show on the slide here. This one, okay, this one, you can just open your existing method with our Shimadzu LC system with lab solutions. You can convert your method, okay, from a HPLC conventional method to a UHPLC fast method. Okay, then after that, you can just save it as a method file and then you run it. Okay, so it's just in a few clicks only, very simple, straightforward. But we also understand that not all users, right? Some users, they might have Shimadzu system as well as other vendors' system. In that case, right, you're probably using other CDS as well. In that case, we have a separate method transfer tool like this one, which I show on the screen now. So for this separate method transfer tool, you can configure any other vendor's system. Okay, for example, be it like from vendor A, vendor W, or vendor T, okay, you can just key in their system delay volume with the mixer volume and the column that you use. You can simply just click this convert button. You can convert to a Shimadzu system. So in the Shimadzu system here, right, you can also configure whether you are using I series or you are using N series. You can just set on the configuration here, and then you can convert the method easily. So two different method transfer program. One is separate program, individual. The other one is the built-in, integrated in the lab solutions. So Anil, I hope I answer your questions. Time is limited, so I want to answer your second question also you mentioned. You are asking the manual, I believe you are an existing AI series user. You say that the manual only mentioned about the minus 12 degrees Celsius for low temp. But we do have an optional. We do have an optional column oven low temperature optional tool that can, if you install this optional tool in your column oven, it allows you to get your column oven temperature, right, to be as low as room temperature minus 15 degrees Celsius. Okay, Anil, I hope you listen carefully, minus 15 degrees Celsius. Okay, yep. So back to you, Vinto.
Thank you, Wan Tong, for the answers for these questions. Hopefully your questions are answered. Now let's have one more question from this person. So let me share. This question is from Mr. Sai Kumabi-san. So how can we develop the configure COA concept with regulatory acceptance? For this question, let us invite Dr. Khani to answer for this question. Over to you, Dr. Khani.
Yes, yeah. Yes, we can use the configured COA, which is a very unique function, which is very much needed for the production who are supplying the drugs for the multiple markets and multiple customers range, where there is a need for configuring the COA in a different format, but the testing is the same sample. And how it works in a compliant way, because when you see the COA, it contains the test parameters, the results, and specification. Here, the results is not changeable. The results is same. What we can configure differently, we can configure the header, we can configure the footer information, and the description, the name of the test, the description of the specification, and display the results as such. In such a way, there is no gap for the compliance because we are not altering the results anymore. The results with the same unit will be displayed in the COA. The rest of the information can be changed. And even it allows you to change the description, doesn't allow you to change the authenticity of the test results. For example, the person who applied the signatures and approve, that information cannot be changeable. So the configured COA is a very unique and very useful function of LIMS. This will allow you to perform the analysis and give the certificate of analysis for the different market in a very, very robust compliance way. Hope I answer your question. Yeah. Please feel free to post the questions in the Q&A chat board. You can get the answers by email with all the questions. Yeah. Yes, Bintu.
All right. I think because of time's constraint, let us conclude this session for today. Thank you once again, Dr. Kani and Wan Tong. I'm sure our audience has learned a lot from your sharing today. Goodbye for you too. To those who have posted questions that weren't answered due to time constraint, don't worry as well, because we will be responding to you via email.
Yeah, Vinto, I think our online audience, they are also asking about how to get a certificate of this webinar. Yeah.
Yeah.
Okay, so
right.
you will also get a certificate after attended this webinar. Yep.
Correct. So before we end this one, we appreciate if you could spend some time to fill out the survey questionnaire on the right side of your console panel. So this will help us to provide better webinar content for you in the future. Also, we would like to just inform you about the certificate, which I think most of you has been asking this question. So if you have the certificate button, you can look at the bottom, the console panel, you can click it and you can download the certificate from there. So if you have any more inquiries after this session and would like us to contact you, of course, please feel free to drop us an email at this marketing@shimadzu.com.sg. But then if you cannot know where is the email, you can just click one of the email widget as well. So it will bring you to the marketing@shimadzu.com.sg. Or also alternatively, you can also contact your local sales representative for more information as well. So with that, I think we can conclude today's session, and have a great day, and we look forward to having you again. Thank you.
Thank you.
Thanks. Bye.
Excellence in science. Shimadzu.